So we will begin. We have got Director Afri with us, and though you cannot see, this is we are sitting in a seminar hall which is full of all the scientists and all the technical officers with us here in the hall, and there are many people connected online. So we will start now with a due permission from Director Afri. Yeah. So we'll start this monthly seminar. We every month we organize one seminar in which we call the experts in the respective areas of research. Dr. Sharad Srivastav, in his 24 years of research career, has made significant contributions in the area of quality control of crude drugs, products, chemotaxonomy, bioprospection, and development of herbal products. He has developed quality standards of more than 150 single herbs and also identified biomarkers for their quality control, which finds place in Ayurvedic Pharmacopoeia of India and Indian Pharmacopoeia. In the area of chemotaxonomic studies on medicinal plants, he has made excellent contributions by identifying quality planting material, that is QPM, and which we also do in research for different purpose for quality planting material of many different medicinal plants, and out of this and which are used as lead source of quality raw material, that is QRM, from QPM identification to QRM of herbal industries. So these are two important things, and he has published more than 175 original research papers with a total impact factor of more than 198.9, and in peer-reviewed journals. He has authored 9 books, and he has, to his credit, 18 patents. 18 patents to his own credit. For his organization, he has been, doing this, and 11 of his patented technologies have been commercialized. Many times it happens that people do have patents, but there is no commercial implication. But for him, 11 of his patented technologies have been commercialized to industry, namely herbal. There's a different products like fruit-based fermented health drink, herbal anti-cough syrup, anti-cigarette herbal formulation as an antidote to tobacco, and the very famous BGR34. BGR34 is an anti-diabetic herbal formulation which is very common. You can find it on any drugstore anywhere in the country. BGR34. That is his product, and he has patented that. And the UR05 product for the kidney stones, anti-dandruff hair oil, herbal hand sanitizers during COVID time. That is what he had prepared, and then herbal decongestant sprays and Ayurvedic immune booster Josh. Besides that, the ladies may be interested in knowing that he has also developed a herbal lipstick. He is honored with a Young Scientist Award from Government of Uttar Pradesh in 2010 by the Sai Ratan Samman 2012, and 6 times winner of the Dr. P.D. Sethi Award. He is a fellow of various scientific societies, fellow of Society of Ethnobotanists, Society of Ethnopharmacology, life member of National Academy of Sciences, India, and Royal Society of Chemistry, London, UK. He is recognized as on national and international forums, and has visited he has world traveled, and his leadership skill, he has handled 38 research projects so far, funded by different national agencies, different funding agencies from India, and different even external funding agencies from foreign countries. And he is a national coordinator of 4 mission projects. So we welcome you, Dr. Srivastav. It's a pleasure to have you. And without much, coming between you and audience, I hand over the mic to you, and the stage is all yours. And we would love to see the presentation.
Thank you,
And
Dr.
hear you.
Thank you, Dr. Tarun. First of all, again, I am thankful to Director Officer and Dr. Tarun for giving me this opportunity. I am going to share some of my field experience and my own observations in the area of medicinal plant research. Basically, in general, it's medicinal plant wealth of country, but you can also consider it as an NTFPs because I came to know that most of you are from forestry background. So what we generally do, we also go to forest for collection, take help from the forest officials all across the country, and then Not only take help, we also recognize in all the sense IP. Even in case of IP also, we do share all the IP, because ultimately whatever we are doing due support and credit from the forest department. So even the challenges or opportunities, these two terms, challenges, I am going to share some of the issues which are of concern to our country's perspective, and opportunities for our scientists as well as the research scholars, where we can do something in terms of R&D, or in terms of value addition, or in terms of commercialization for the country as well as for our own credit. So, Dr. Tarun, slides are visible and I am audible. Dr. Tarun,
Yes, slides are visible and you are perfectly audible,
or
loud
then,
and clear.
yeah, because sometimes we
Perfectly going on well. Yeah.
okay, so let me share some of the positive points of our country. Our country is enriched with so many diversified, can say ki, wealth, because we have almost 20 agroclimatic zones, and we are one of the 7 mega biodiversity countries. 7% of the whole biodiversity we cover in our country. If you see in terms of flowering diversity in our country, we have more than 47,000 flowering species available in our country, and about 9,000 species are used in Indian systems of medicine and folk. We have a long history of usage of medicinal plants. Ayurveda is one of the texts which is available with us. It's an old 5000 BC old text. Even more than 2,000 species are now used in modern medicine. And out of these 9,000, around 1,200 species are in trade, of which 240 plus with consumption of more than 100 metric ton. 40% of species in high demand sourced through cultivation. Still, we have scope for cultivation. We have a large network of institutions, universities involved in medicinal plant research. And currently, we are having export potential worth around 500 million US dollar, which is 2.5% share of the global market. And the last one, we have more than 9,000 units engaged in manufacturing of herbal products. So this is the strength of our country. We are mega biodiversity country. We have a rich heritage as well as variable agroclimatic conditions, and rich repository of medicinal plants. If you see this map, all across the country, from Himalayan or trans-Himalayan zone, marine or even desert or even rainforest ecosystem, we have a varied diversity, and even not only varied diversity. Sometimes one species which is growing in Gangetic Plain is available in arid zone as well as in Deccan Plateau. So see the diversity pattern and availability of medicinal plants all across our country. If you see the demand of medicinal plants in different systems of medicine, Ayurveda cover around 1587, Siddha 1128, Unani 500 plus, Soa Ripa is another emerging system of medicine which was earlier known as Tibetan system of medicine. Even in homeopathy, if you see, we have around 450 medicinal species used, and in Western system of medicine, we have 192. This data is now updated. We have more than 250 species available in Western system of medicine. If you talk about the supply of medicinal plants for product development, this is as per NMPV surveyed data, that 242 plant species recorded in high annual trade, and out of these species, still we are dependent on forest for more than 114 species. From Himalayan forest 36, tropical forest 72. From the wasteland areas, we have around 59 species. Cultivation, which is only 40% of the total demand, 54 species, and around 15 species still we are importing from other nearby or other countries. So these slides, what I mean to say that still we have scope for different kind of research activities, either in a as an individual scientist or even in consortium mode or in network mode, we can do a lot for our country. If you see the potential of India in medicinal plant sector, yes, through cultivation of our medicinal species, we have remunerative substitute to traditional crops. Suitable medicinal plants are suitable for intercropping, multi-story cropping, or even agroforestry, which generally even forest department also do. We can also utilize barren and wasteland. And nowadays you see that even our honorable Prime Minister and Minister also, they are emphasizing on working in northeastern part of our country. So it may be a game changer for northeastern states which are affected by the Naxal movement. And of course, we can become world leader because of having advantage of variable climatic conditions and rich traditional heritage and knowledge, as well as scientific capability and potential of our own Indian brain. Even small or marginal farmers, they have an alternate cultivation strategy. Sometimes even which we have seen in many areas that small farmers, they are growing tulsi and selling it and earning a lot of money. Even makoy cultivation is going on. So I mean these are the potential or opportunity areas where we can think over, or we can make a kind of game changing attitude. But even though what is happening, अच्छी अच्छी बातें तो हम लोग कर लेते हैं, बताते हैं सब कुछ है, but when we are going to work on medicinal plants, still we are confused what to do. And why? Because the challenges are still existing: scarcity due to depleting biodiversity, adaptation substitution, the major cause, inadequate cultivation, and then indiscriminate harvesting from natural sources. So these 3, 4, or 5 challenges are creating opportunity. And if you see, still we have only few SOPs for commercial cultivation. I mean, agrotechnologies for only few medicinal plants available in our country. So we have an opportunity for development of agrotechnologies and SOP for judicious collection of from natural habitats, and chemo profiling for identification of admixtures. Although our regulatory bodies, they are doing a lot, getting support from different R&D institutions, but still we have opportunity. And then protection of therapeutic RT species. This is also an area where many agencies are trying to provide a lot of funds to work over. And another important opportunity is identification of QPM, quality planting material, or QDPM, quality defined planting material, which leads to QRM for industry, quality raw material for industry. So I'll give you some of the examples, start from adaptation substitution to QPM, even in how we are going to product development, from getting natural products effectively available for societal need. So I'll take few slides and I'll give you some of the live examples which what is exactly happening. Although as I mentioned that we have regulatory guidelines in our country, our Ayurvedic Pharmacopoeia of India, which is from Ayurvedic Pharmacopoeia Commission, and then Indian Pharmacopoeia published through Indian Pharmacopoeia Commission. So these two regulatory bodies, they time to time publish their different volumes which contain the quality standards and quality parameters for single herbs, even extracts, or even phytopharmaceutical ingredients, or even SMPs or SOPs for formulations. So, I mean, these are the government gazette which is available. I am sure our learned scientists, those who are listening this lecture, most of them, they know that these are the regulatory guidelines which is available and it is in public domain. But time to time, it is also needed to be revised. So we are also helping them for the revision, because in few cases, more than 10 or 15-year-old data is there. So it's mandatory now to commission that to revise it, and we are also helping in it. Now coming straight to the regulation and substitution issues in our country. You know, in market, when you go and you ask someone, the shopkeeper, that to provide Brahmi, so he'll ask you which Brahmi you want, Neer Brahmi or Manduk Parni. So if you're not aware of that, which is the Brahmi which exactly you want, you will say, okay, give me any of these. So these are the problems which you are facing. If you ask Atish, which is an aconitum species, you don't know which aconitum species you are getting. There are 6, 7 species available in market in name of Atish. Like Vidarikan, this is an ingredient, a key ingredient of Chyawanprash. There is a lot of confusion and adulteration or substitution happening in the market. So I have categorized these adulteration substitution in two subcategories. One, undeliberate, which is done unintentionally or sometimes deliberately because of earning a lot of money out of it. So undeliberate is of course because of confusion of common name, same plant with different names, or misidentification due to similar morphology, and lack of skill for identification, because nowadays like our RET species, our taxonomists are also a threatened species of RET species. So in our country we are losing taxonomists. So or even field botanists. So that is why have given due credit to our forest officials, because they are very much helpful to us for the field studies and even sometimes helping in identification of plants. And then season and stage of collection is also very important. Sometimes in wrong season, if you have collected and supplied to a industry, you will not get a good quality raw material, and which will not lead to a good or efficacious product. Diurnal variation is also an important issue, which is happening through undeliberate adulteration or substitution. So undeliberate, if you see, Punarnava, we all know that it is a Rasayana drug. Officially, Borhavia diffusa is known botanically for Punarnava. But when we see, we have these 4 species available in name of Punarnava. If you go to market, you may find Borhavia diffusa or even sometimes Borhavia repens or erecta, or in many cases we have observed Tanthema portula castrum, which is not exactly Punarnava but morphologically very much close to Borhavia diffusa, and that is why it is easily sold in market. If you see morphologically Borhavia repens or even Borhavia diffusa, this Tanthema also looks like similar to Borhavia. So this is what unintentional adulteration is happening, because we don't know the taxonomy of this Tanthema and we just collect it because it's morphologically similar to Borhavia. So these are the issues which is happening in when you go to collect and or when you procure it from the market. So I was talking about another important adulterant for Vidarikan. Vidarikan, if you I hope many of you know this plant. It's a Putil botanical species from the family Fagaceae, and one of the key ingredient of Chyawanprash, and it contains purine. Purine, and it is one of the best Rasayana drug. And nowadays this Vidarikan is now used for this what you call that हमारा जो मद्य निषेध में होता है अल्कोहल को डी एडिक्शन के लिए, उसके लिए इसको बहुत यूज कर रहे हैं. Although this is one of the important herbal drug for reproductive health care. So what is happening? This Pyrrheria tuberosa it grows in foothills, and when you see the morphological structure of Pyrrheria, it is a tuberous root. It's a bean-shaped tuberous root, and it grows inside the rocks. So we have only 2 options for its collection. One, either we can blast the rock and then we collect, or we wait for landslide and then we can get. Because if you see the demand of Chyawanprash in not only in India, world market, and as per demand of Chyawanprash and as per the percentage required of Vidarikan in that Chyawanprash, it is really very, very tough to get this raw material in sufficient quantity as per industrial demand. And what is happening is, there is another tuberous species, Ipomia digitata, which is very common in almost more than 60% part of our country. If you go to southern India, you will definitely find this Ipomia digitata only. And because in market, product market, you will not get a fresh one. So what is happening? They chop it, and you see the pieces, they are morphologically almost similar. Pre-image, pieces. And surprisingly, this Ipomia digitata is not at all similar to Pureria tuberosa, neither in terms of morphology nor in terms of chemistry. So this is what is happening, and we as a pharmacognosy division in CSR NBRI, we are having a MOU with both these regulatory bodies to resolve such kind of issues. So we are working on regular basis for identification of adulteration and substitution, and we are providing the details to these two regulatory bodies. You know Ashoka. Ashoka wrist is a very common Ayurvedic preparation available in market, and Ashoka is also a kind of plants which is known in our Indian systems, even in Hindu rituals. If you see the official drug Ashoka is Saraka Ashoka, Sita Ashok जिसको कहते हैं. But in most of the places, we know only this Polyalthia longifolia is the Ashoka, which is not at all the Ashoka. Even in roadside or even in our home boundary wall, we plant this Polyalthia longifolia as an Ashoka tree. But officially, this Saraka Ashoka is the genuine plant sample, which is supposed to be used for preparation of different Ayurvedic formulations. When we surveyed the market, I am mentioning only 4-5 species here. I was shocked to know that almost more than 11 species are used in name of Ashoka. I am just showing few of the examples like Bauhinia variegata, Schweria robusta, or Polyalthia. Saraka, 2 species are available, but in majority of the market we have only Polyalthia longifolia. So this is what is happening. And if you analyze in lab, do its chemoprofiling through either any chromatographic tool, HPLC, HPLC, or even we have now many other tools and techniques, UPLC, or you go for NMR or any other higher analytical tools, you will find out that all these species are entirely different. They are not at all matching in terms of chemistry or even efficacy. But this is the reality of our market, which time to time we are checking, and we are providing the details to both these bodies for regulation through their different sub-agencies. Even Sida species, very common is Sida acuta or rombifolia. Sida in Ayurveda known as Bala mool. Bala means Balvardhak. And if you see Ayurveda text or our government gazette, Sida cordifolia is the official drug designated to Bala mool. And I am sure your field botanist or your first officials or the scientists know this. This Sida cordifolia is very common in Rajasthan and Gujarat area, and some part of UP and Madhya Pradesh. But in rest of the part, this Sida cordifolia is not at all available. So what is happening? Even if you go to Delhi market, even if you go to Tuticorin market, you will find out other Sida species are available in name of Bala mool. And majorly Sida acuta or rombifolia or another South Indian species is Sida retusa is available. But Sida cordifolia is not at all sufficient enough to supply, make a supply chain to industry. We analyzed all the species, almost 9-8 species we have analyzed. What we found, all these species they have a quality marker ephedrine and pseudoephedrine. We have analyzed based on these two markers, and luckily all the species contain ephedrine and pseudoephedrine. And we suggested these regulatory body APC that to include all the species because we have scarcity of supply, so we can include other species too, so that our Sida cordifolia get conserved, and we are not going to lose this species in name of Bala mool. So now at least it got accepted that other Sida species will also be used in name of Bala mool. एक और इशू है मिस आइडेंटिफिकेशन का. इफ these pictures are properly visible to you, Jatamansi is very common Himalayan raw drug. If you see Jatamansi original, this is the original Jatamansi, and just parallel to it there is another morphologically similar looking raw drug which is botanically equated to Selenium vaginatum. is also sold in name of Jetamalsi. Surprisingly, entirely different in terms of metabolites, even in terms of efficacy, but it is available in market. And same is the case with Texas Policiana, because Texas is very common. Known to all the people because of Taxol and its anti-cancerous leaves. So if you see Texas Policiana needles, there is another species, Abies spectabilis or Abies webiana, which is very common and adulterated in Texas Policiana just because of having similar morphology. And it is happening because of misidentification, because the traders, they are not going to collect, and even we as a botanist, we are not going to collect in bulk for industry. So unskilled people, they are going to field or forest or associated site for collection. Sometimes they know, but sometimes when they see another plant with similar morphological characteristics, they collect this. So this kind of misidentification are also happening, and because of that, we are not getting the proper or quality raw material. Coming to seasonal variation, yes, of course it happens, and this is one of the examples that Rhium himoide, it's an Himalayan plant, rhubarb. If you see in winter season, there is no anthraquinone, it gets inactive, and it gets oxidized and available as an active ingredient during the summer. So if you are going for collection of any plant species, even for your research activities, please be careful of its season, its maturity, and as well as stages of collection. I am just sharing summary of these stages of collection. If you are going for collection of these, be careful. You try to collect just before flowering starts. Flower when it is fully open, and then rhizome or root when plant is fully matured. So these are the basics which are recommended for any kind of collection strategy, and for research as well as commercial prospection. So there are a few of the examples which I have mentioned here. Although this presentation will be, I think Dr. Tarun will be available, and if anyone wants, you please share with them. एक और इशू आता है substitution of exhausted product, and this is the best example which I always share: the clove and cardamom. Not good to say, but almost all of us have visited once or twice or even frequently to any pawn shop and any pansari shop where we procure spices for our kitchen. So we have two contrasting examples here. When you go to a pansari shop and procure something for your kitchen, sometimes you get a very good quality greenish look cardamom or very golden brown colored clove. And same when you go to any pawn shop, you see एक बेचारा मरियल सा सूखा सा दिखने वाला इलायची दिख रहा है, और similarly this clove is also there. See the morphological difference. So what is happening is traders, they are doing partial or distillation. They are extracting this VOC content, essential oil, and then the remaining part after drying, mixing with these two genuine, or even sometimes what they are doing, they are spraying the VOC content on these two product and selling in market. So we have 2-3 category now in market, A, B, C. It comes in C or D category depending upon your need. So this is what is actually happening in the market. So now what to do? Although government gazettes are there, we have WHO guideline, we have our own NMPP published guidelines for good collection practices, good quality control practices, GLP and GMP practices. We have all sort of guidelines. Let me share a few of the slides in very short. That this is the first step. If you don't know anything about your raw drug, you consult any of the drug repository which is available in and around you. And we too have the similar kind of drug repository. We have more than 2,000 authentic drugs which we collected ourselves and displayed in our this drug repository. This is not only a kind of display item. We also serve different agencies for creating the clearing the dispute happening either in narcotics or even in excise or even with researchers from institutions or universities. Another point I wanted to share here, Dr. Tarun, that suppose, particularly if I say that AFRI is working on a plant called Ashwagandha. Ashwagandha is very well known in Rajasthan. Nagaur is the place where it was well known and recognized because of Nagauri Ashwagandha. Nowadays, we have many more such locations where we are getting good quality Ashwagandha other than Nagaur, even from Rajasthan. Whatever collection your scholars or your scientist colleague or even any of the audience, those who are listening, are doing any collection, one should prepare a passport data sheet. Now it is a Government of India recognized passport data sheet. It was prepared in consultation with ICR, CSIR has prepared, and now we made it mandatory for all the researchers, those who are working in the area of medicinal plants. Here in this passport data sheet, you have, I'll provide the specimen copy if required to any of the audience. The importance of this passport data sheet is if you have collected any plant, suppose from your Barmer area or even Khudi area, because Khudi is also well known for different species of Tribulus. So suppose if you have collected Tribulus domigenosus from that area, and you have documented all the details, its availability, its abundance, its GPS coordinate, stage of collection, amount of collection, even similar looking other species available in that area, soil details means soil morphological details, and prospection in terms of agronomy, in terms of any traditional knowledge or any other field note. And the collector who has collected, he also need or she also need to sign and then authenticated by either by himself or herself or by any other expert. This data is mandatory nowadays in terms, suppose if we are going for bioprospection, we should have such passport data sheet, because after bioprospection, when you go for commercialization, commercial issues comes and NBS made mandatory to have such kind of information. They also ask when you submit any patent and NBA clearance. So this is a kind of information which is nowadays essential to keep on record. So I always suggest, whenever I make any such presentation, I suggest just to have such kind of information available with you. And Article 8J of benefit sharing is based on this. The Kani tribe, on which Dr. Pushpangan has worked out and developed a drug from Trichopas jelenicus, commercially known as Jeevania, for which he got this Equator Initiative Award. And then this Pushpangan model of benefit sharing was established in Earth Summit. Because of all these information and involvement of tribal community, Kani tribe, the benefit sharing model was established, and the commercial as well as other benefits were equally shared with the Kani tribe. So this is a perfect example of benefit sharing, Article 8J. So if you have all sort of information, you have to mention the person who has given you the tribal information or the tribal recipe as well. His details, his contact number, and all that you have to document. So that is what the importance of this passport data sheet. Of course, if you have anatomical details, it is a very, very basic parameter, just a kind of baseline information. One can easily do, even class 12 standard or 10 standard candidate can also do. And what we are doing, just I wanted to share here that we are preparing kind of anatomical atlas, not only anatomical descriptors, along with anatomical descriptors, we are also providing chemical profile, either through HPLC or even HPTLC, because nowadays HPTLC is very cheap and economic to any person, those who wants a QC data of their raw drug. So we prefer to develop HPTLC profile. So this kind of atlas we have published we have already published one. This was released by DG or the Honorable DG CSIR, and this contains only very, very basic information. And even I tell you, AFRI can also join hand with us in this case. It's very, very simple parameter. And nowadays, even APC also required, and they are also parallelly publishing atlas of anatomical atlas. So here your botanist can also serve for preparing the anatomical descriptor of any medicinal plants of your arid zone, as well as your chemistry person can contribute in providing HPTLC or any other chemo profile of the rotor. And with that compilation, you can also publish, or you can take help from NMPB or Ayush. Also, they support for such kind of activity, and you can publish your own compendium or atlas. One thing, just I wanted to share you with all of you, that whatever slides I am sharing, I initially told that if you have any query, you please feel free to ask. And I tell you one thing very proudly, that whatever slides I am sharing, each and every slide had and have worth in terms of publication or in terms of recognition to these two regulatory body, or even in terms of IP or commercialization. And I'll share in the last how we are earning out of it. So HPTLC is a perfect tool, very simple tool by which you can go for the quality control of product. And Berberis, or in Ayurveda it is called Daru Haridra, is the best example. Uh, just I want to share that Daru Haridra botanically equated to Berberis aristata, officially recorded in our API. But the issue is that over exploitation of Berberis aristata is now created a kind of scarcity of aristata. I am damn sure, because I have worked a lot on different Berberis species. I can give you the details on fingertip. Nowadays, Berberis aristata is not at all available in natural population as per our requirement. So what is happening is Berberis aristata, Chytria or Lycium, all these species, not only these three, we have many other Berberis species in southern part, Berberis tinctoria, even Mahonia or even Posinium fenestratum is sold in the name of Daru Haridra. The most interesting part is that whatever the previous data was available, that Berberis aristata is the best source of berberin, which is an isoquinoline alkaloid. When we analyzed, we found that the maximum content was available in Berberis chytria. This was really shocking to us even. So we suggested our APC, Ayurvedic Pharmacopoeia Commission, to include all these species, because all these species not only contain berberin, even 4 major isoquinoline alkaloid is very common in all the Berberis species, not in even root as well as their stem part. So if you see the market, you may find in most of the market we have stem samples mixed very frequently and sold in name of Daru Haridra. Stem means stem of Berberis species. So that is why we worked on it. And as I said, that every slide has worth. You won't believe that by this plant we have published 9 papers, and one process we have already commercialized to one company for small 2-step process for commercial berberine. I mean isolation of berberine. So this is very small 2-step process we have developed, and if anyone want, I can go in detail after this presentation. So another tool is HPLC. HPLC is also very useful in terms of uh, developing quality kind of parameter of any formulation. So Trifla is one of the example where we worked out, and we have provided the details how to uh, make a quality control SOP for any Ayurvedic preparation. Nowadays, another chromatographic tool is getting popularity, which is called as OPLC, over pressure layered chromatography. It's a hybrid instrument, and in offline mode, it works as an HPTLC, and in online mode, it works as an LC, liquid chromatography HPLC. The advantage with this particular instrument is that suppose if you are analyzing any plant sample for its metabolite, say for even berberine or even shatavarine or bidaferine, you can you can collect your peak, and you can have your own library of marker. And not only that, if you go in deep of through this particular instrument, I am sure that you can also develop PRS, phytochemical reference standards, which leads to CRM, certified reference material, because in our country we are running problem of getting these phytochemical markers. still we are importing from different countries. We don't have our own setup here where we are getting CRM or PRS. So PRS now, National Remedies, Amitabh Agrawal Group, they have started, and we have almost 70-75 known metabolites available through them. But still CRM in our country, we don't have any supplier. Even no institution, they are having their own CRM. Fortunately, CSR has initiated one such mission, and our lab recently got recognized for 7 CRM. So we are providing CRM. This is still in process. We are not going for its commercial prospection, but still it's in process. Now Indian Pharmacopoeia Commission, they adopted our this CRM, and now through IPC it may get available to any institution. So this OPLC is also a simple tool by which you can go for resolving any conflict. This is another area where we can work on NTFPs or high value species, which is available in and around forest area, because uh, if you see, we have certain restrictions as per guidelines, but yes, we have certain areas where we can suggest the inhabitants for its cultivation. So if I go for thinking of alternate strategy for any RET species, so we can adopt such kind of strategy in or nearby forest areas, whether it is in Himalayan zone or even foothills or even Gangetic plain or arid zone. So I'm just sharing two examples here. We already worked out on Daruvaridra, which got accepted in IAPC. Another is that wild yam, Dioscorea deltoides, which is well-known source of diosgenin, natural diosgenin. So now Dioscorea deltoides, although now it is now in cultivation in different places, but still in high, because of high demand, different Dioscorea species are used. We had no alternate strategy. We initiated work on a species called Costus speciosus. It is very common around roadside, even in marshy places, and having huge biomass of its rhizome, which contains diosgenin. So we analyzed, and we have already established the details, and we have forwarded it to APC for its inclusion. So alternate sources we can also think of, we can work on, and in this area even NMPB is providing a lot of fund. No, nowadays Ministry of Ayush and NMPB is having a lot of fund to support such kind of activity, alternate source, QPM, QRM. So as our scientist colleagues, those who are listening, they also need to get support, financial support from different agencies, not only from ICFRE, not from your own institute fund. We should also think of getting financial support from other agencies. So NMPB is very helpful in such cases, alternate source strategy, and providing alternate source for high value medicinal species. This is another issue which Dr. Tarun and myself, we were talking about, location specific cultivation of medicinal plants, or NTFPs. Although NTFPs, it is very common term for the forestry people, but we are restricting ourselves to few medicinal species when we talk about the NTFPs. Here, my suggestion and submission to all the eminent experts, or even to Dr. Officer, that because you are an IFS officer, you know much better than me that where we can think of adopting such kind of strategy, location specific cultivation. So what? I'm sorry. Is there any Dr. Tarun? Am I audible?
Yes, you are audible. Please go on.
Okay.
No problem.
So what is the advantage of location specific cultivation? Because best suited location for it will be useful for getting high concentration of metabolites, desired metabolites, and this kind of promotion of cultivation in a location where it was having optimum growth as well as metabolite will provide QRM, quality raw material to industry. If we talk about the location specific cultivation, yes, that local people will get kind of employment or engagement when we are talking about the commercial cultivation. It will also help in conservation. As I mentioned that there is a species of Tribulus terrestris available in your this border area, Doniginosus. Now what is happening, it's because of extraction of these species. I visited few months back to that area. I was surprised to see, I mean, in the previous season, it was not so common in that area. Although when I visited 3 4 years before that, it was very plenty there. Surprisingly, this time it was not. So what is happening is the conservation strategy is just only to protect or to make a buffer zone. It will not serve the purpose. We can also go for providing options for extraction, whether in terms of whether it will be in case of tribulus or any other species, but we should provide them either through cultivation strategy or even extraction from natural sources. We should have such kind of strategy. So in that sense, it will be helpful, and not only helpful in terms of cultivation, it will be helpful to industry for providing a quality or efficacious product to our country. Now coming to strategy of how we can go for identifying QPM, quality planting material, which initially we talked about as a chemotaxonomic studies. Uh, I just wanted to share one of my personal experience and proud moment. In 2006 and 7, when we talked about this chemotaxonomic approach and identification of QPM, no funding agency appreciated that. I told even that this will solve the purpose of depletion of our taxonomist. We need to modify, we need to adopt modern tool, and our taxonomists, those who are not having good quality publication, by this strategy they can also get good quality publication, and of course they will also get such kind of numeration either through publication or even through IP. So then suddenly in 2007, I met one ICR official. I was invited to NDRI Karnal to make a presentation there. I shared some of my slides and then he asked me 2 3 points. He said, आज के ज़माने में ये एनाटॉमी कौन करता है? ये आपकी रियल स्लाइड है या किसी बुक से कॉपी किया है? He was such a senior officer, and it was really very shocking for me that he asked such kind of question. I said, sir, it is our own. And he said, ये फील्ड वर्क का जो फोटो दे रहा है, ये कहाँ का है? खुद किया है कि किसी का कॉपी किया है? I said, we generally go to field. Then I shared few of my experience with him about the variability issue, that we are not getting optimum concentration of metabolite when we collect it from Lucknow or even sometimes when we collect it from Pali area of Rajasthan. So we had a chat with him. Then he offered me to come with him to Delhi in his own car, official car. On the way, we talked about many things. I showed some of the examples on the way while stopping the car. And when we ended our journey in Delhi, he told me to prepare a concept note on it. So sometimes आपके जिंदगी में भी ऐसे U-turn आते हैं. And that day, we had prepared a concept note. It got approved. And you won't believe the first project which we got sanctioned on this kind of study was that time in 2008 was 4.79 crore. So thanks to NAIP, ICR, who supported or who initiated when given opportunity to initiate this kind of study. And now, last year, MMPB has signed MOU directly with our group for this QPM. So we have already identified 14 high value species and their location specific QPMs. So this is the kind of earning which we got it through this initiation, and still you see only 14 species has been worked out extensively. The strategy is very clear. You have to go to field, you have to map the whole country in terms of its phytogeography. When we are talking about any medicinal species, you have to collect from different parts of country. It doesn't matter whether it is available in Jammu Kashmir or not. You have to go and see it physically. Not only consulting flora will serve the purpose. There is a clear-cut migration in plant nowadays. I am just sharing one example which I'll share in the next slide, where I'll prove that this migration is happening. And then after mapping and collection, you have to bring the samples in desired quantity, raw material, as well as the live specimen along with soil, rhizospheric soil of that area. So these three items are needed when you start your such kind of journey: live plant material, product sample, and soil sample. So live plant material you have to conserve in your conservatory, and then product material after processing you have to go for its a sort of parameters which we have already established through consulting all the funding agencies, ICR, NMDB, Ayush and all. We have made a comprehensive set of parameters for such kind of study. So you have to analyze those parameters, you have to develop the data, and then you have already conserved your collected sample. You have to go for its Q generation studies, whether these collected or conserved samples in our location is having any kind of variability in metabolite content. I mean, is there any upregulation or any downregulation? Of course, when you conserve it in different geographical condition, definitely metabolite will be variable. So this we have already observed. Then thereafter, what is happening? When you see that your Pali Rajasthan sample having suppose 0.3% of etherferine, when we grown here in our condition in Lucknow is having 0.01% of etherferine. So entirely different. But surprisingly, the same sample which we have collected from Pali, when we go for multi-location trial and cultivated in Gujarat area, say for Maheshana area, we are getting 0.35% of etherferine. So it means it is very, very clear from this field observation that we have variability and we can go for location specific cultivation. So first and foremost requirement is mapping, collection, and then analysis and conservation. So after analysis, you will get which is the best locations where you are getting higher amount of metabolites. So by that lead, you can go for multi-location trial so that you will get confirmatory test done that yes, this is and as well as sorry, I missed to tell you about the soil. So soil, rhizospheric soil also you need to analyze in terms of its physicochemical parameters, which will be a correlative factor. Although you can go for both biotic and abiotic parameters, but abiotic parameters only solve physicochemical parameter as well as its elemental analysis will initially serve the purpose. So by that read you can establish a correlation and you can go for prediction analysis, which are the best suited locations for its location specific cultivation. So Tribulus is one of the example which we have surveyed and mapped the country. Although we mentioned here 41, this we are again revising now because it's a 2008-9 started journey. This is the sorry, this is the first plant which we worked on identifying QPM. So we tried two things here. One, Tribulus is well known for prototribulin, protodiazine, tribulosine, and rutin, and all these four phytochemical compounds are well known for its different efficacy. Either it is protectile or aphrodisiac or testosterone or even anti-inflammatory activities. And the interesting part that after our study, start from Leh Ladakh to Kanyakumari, we found that arid zone is the best suited location for Tribulus terrestris particularly or even other Tribulus species, which contains optimum amount of metabolites. And here another advantage which we got out of this study that we have identified customized chemotype. Say for example, in case of ashwagandha, you all know that we have 4 chemotypes already identified and recognized. Here also we got 4 chemotypes, one which is rich in protochemistry. So nowadays when we talk about the phytopharmaceutical drug mode, so in that case this kind of study is very helpful, that we have customized chemotype. If suppose any industry want a tubulin rich accession, we can suggest them that to go and you can collect or even cultivate or go for contract farming in Jaisalmer area, which is a good source for tubulin rich chemotypes. So this kind of strategy is really very important and helpful, and your institution particularly will play an important role for such kind of activity where QPM is required. Because you are having AFRI there, and even Kazri, ICR Kazri is there. So they have also, I think, some made some initiation in this area. This is another example, Gloriosa superba, known for its metabolite colchicine. Initially, I mean, long back it was banned by FDA. Now revoked the ban. Now its demand in world market is increased like anything. See, even from our country, majorly Italy is the largest buyer of Gloriosa superba, and I'm sure that our scholars, they know that Gloriosa is the state flower of Tamil Nadu. So it means that Tamil Nadu is the well-known place or the most loving place of this particular species. Its tubers are highly important in terms of product for gouty arthritic conditions, and seeds are of course used for isolation of colchicine, which is used in inducing the polyploidy. So Tamil Nadu is the key location for cultivation of Gloriosa. Yes, there are various farmers cultivating this particular species in that area, start from Coimbatore to Tirunelveli. But when we surveyed the whole Tamil Nadu, its peak season, we were surprised to say that in natural habitat it is very, very rare to see. So I thought to move towards Kerala. From Madurai, I moved towards Munnar, and soon when I entered from Munnar, when I entered downside, I was amazed to say this particular Gloriosa superba available in plenty with huge amount of biomass. जो इसके वी शेप ट्यूबर्स होते हैं, अगर तमिलनाडु में 4 इंच और 5 इंचेस के थे, there it was around 6, 7 or even 8 inches, and good quality tubers. So I was really very surprised how it is happening, because Tamil Nadu is the main location for Gloriosa, either from its natural habitat or even from cultivated field. And soon when I entered Kerala and traveled the whole Kerala, I found a clear-cut migration of this species from Tamil Nadu to Kerala. So migratory weavers are still existing even in plants because of their stress conditions, and even sometimes there are other abiotic or biotic factors which are playing important role for their survival as well as the quality of material, whether it is planting material or raw material. So here in case of Gloriosa, we mapped the whole country, around 130 plus collections we made, and then we identified the area which is the best suited for its cultivation as well as having optimum concentration of colchicine. Of course, as I mentioned that Kerala, we found big rhizomes, this are good quality raw material. In our study, we found that Wayanad is the area which is having high content of colchicine in its tuber, 0.86%, and this is the first ever report in world. So researchers, I tell you, this particular plant, 137 accessions of Gloriosa superba has given us 13 good quality publication. Start from chemistry, biodiversity, industrial crops and products, even to Royal Society advances, and many more. Even phytotherapy or general vectopharmacology. To see the such kind of study, how much you are earning. Not only taxonomy you are learning, you are earning in terms of your candidature. Good quality publication and a good funding support from agency like ICR, who is really very tough to handle. We got it, and then finally when we got this good, lead sources or location specific sources, We worked on its a kind of product development. I'll share that example in the last slide. That by that lead, we have developed a product which is of really great value to us. And then, in fact, Themis Pharmaceutical has already given LOI for that product. So, see the potential of your research, particularly in such areas where we are never thinking of going to adopt or opt how to work. Because it's a field-based study, you have to go 50-50, you have to go to field, you have to collect, you have to follow a holistic approach, judicious collection strategy, then judicious analytical strategy, as well as interpretation or correlation of your soil parameters along with the bioactive markers which you have quantified or identified, and then finally for its commercial cultivation strategy like multi-location trial or stability studies, and then identify QPM or variety even. So we have already developed one variety out of it. This is another plant, Coleus phoscoli, which is well known for its dieternate phoscolene. Here we have mapped the whole country, and then of course western coast of Malabar is the best suited location. First ever report, as in case of Gloriosa, similar in case of Coleus, we have reported 1.53% of phoscolene, and this plant also given us a lot of recognition, around 11 publication, good quality publication out of it. Centella asiatica, you all know, it's a memory enhancing plant. We worked on its four biomarker: asiatic acid, metacasoside, asiatic acid, and asiatic acid, asiaticoside, metacasoside, and metacassic acid. And we have already identified the sources. We have mapped the whole country, 178 accessions we collected. We mapped the country, we quantified following the same protocol as I mentioned, and then finally we have identified 4 chemotypes. This is a common weed. I just wanted to share with you people. Sometimes हमारे लिए कूड़ा भी बहुत अच्छा होता है. हम इसको कूड़ा या खरपतवार कहते हैं हिंदी में. But you won't believe that this plant also given a lot of reward to us. We were working on identifying a natural supplement for controlling the ticks in cattle. We were screening around 100-200 plants. Out of that, we got 2 leads. One is ageratum, another is a different species. And then when we started working on it, surprisingly, this is really a wonder plant. We have developed ultimately a product out of it, and we have transferred this technology to Ajay Biotech, a Pune-based company who is already in business now with this particular lead. And I can say proudly that this is the first product in world global market which is a purely herbal product or herbal acaricide, because earlier to this we were using synthetic pyrethroids to control ticks, which are causing a lot of cancer issues in our cattle, leading to the milk which we are drinking is having similar toxins. And in Punjab, you all know that there is a district which is known for cancer patients, and there is a train running from that district to Mumbai, particularly for Tata Cancer Center, which carries a lot of patients to Mumbai. So see the problem where we as a researcher, we are trying to go for the reverse action, and accidentally when you follow a holistic approach towards your research, you can lead or you can end up with a good quality product and national as well as international recognition. So what we did when we developed that product, so industry also wanted raw material, and this plant is not at all officially available in any commercial market. So then we first analyzed, then we developed all sort of parameters, then submitted to both the commissions that to include it, although it is a weed, but since there is a product out of it, so you can please include. So it's in process of inclusion, but we mapped the whole country. We identified the sources based on leucosin content, leucosin 1 and 2, and then caryophyllene oxide. And then, as I mentioned that Jetamansi, we have already identified 2 chemotypes, which is in cultivation. We also developed cultivation SOPs for these species. This is another plant, Costus speciosus. We have mapped the whole country, and central India is the best suited location for this particular species. Then why I am just focusing a lot and emphasizing you for cultivation? Because ये दर्द मेरा मेरे से बेहतर आप लोग समझेंगे जो फॉरेस्ट्री के हैं, जो फॉरेस्ट बैकग्राउंड से हैं, उनको मुझसे ज़्यादा अच्छा या दर्द इस बात का फील होगा कि जब हम लोग एज ए रिसर्चर जाते हैं फॉरेस्ट में, तो हम लोग भी कभी कभी खुशी में बहुत ही मतलब वेग तरीके से कलेक्शन करते हैं. So what we are doing, we ourselves is decreasing our wild biodiversity. If we compare it with cultivation, yes, of course, we are increasing, we are conserving and increasing. Coming to identification issue, in cultivation there is no issue because we are cultivating a known species, authentic species. If we talk about supply, suppose if I have collected any plant from desert area, and the second time when I am going to collect it, it may not be available. So supply is another issue. In cultivation, you have controlled supply. Quality, you are not sure about which quality raw material you are getting from a wild collection. Cultivation, you have a known quality. Genetic improvement, yes, of course, in cultivation you have an opportunity. Agrotechnology, yes, profitable है. Then post-harvesting, post-harvest handling. In cultivation, you have an optimum SOP for post-harvest management. In terms of adulteration or substitution, for the cultivated material, it is very clear that this is safe. You are not having any kind of adulterant or substitute or even foreign metals. So I've just passed through few slides where not only NBRI, some of our other labs, they are working on it. So this is example of cultivation strategy where the cultivation cost is around 1,64,000 per acre, and net profit is around 5 lakh rupees in terms of per acre for Gloriosa superba. Similar is with Costus, then Centella, then Chlorophytum, then Mushdana, Avis Moschus, then Pach, Eporus calamus. Ashwagandha already is very, very established agrotechnology. Then Kevach, then Shatavari, which is very well known, established SOP. Then Isabgol, the most traded plant species. Then Andrographis. Then Curcuma also. Curcuma nowadays getting a lot of attention in intercropping. So here you see intercropping or even in forest areas, you have an opportunity for such kind of strategy to get implemented. So not only curcuma, there are many other species, gingiber species, many other herbaceous material which are nowadays in practices for intercropping. So not only curcuma longa, we have many other curcuma species or even varieties. Even we are also providing technical backup for such kind of strategy. We have our own developed one variety of curcuma which is having more than 3.5% of curcuma. So intercropping, through intercropping you can go for many such gingiber species. Aloe vera, it's very common, you know. Now coming to how after all these research we are going to develop a product. ऐसा नहीं है कि हम लोग सिर्फ प्लांट सैंपल्स लेते हैं, उसको खूब पीस के मिला करके कोई भी आयुर्वेदिक प्रिपरेशन बना देते हैं, जो नॉर्मली हमें ऐसा फील होता है व्हेन वी सी आवर प्रोडक्ट्स व्हिच इज अवेलेबल इन एनी शॉप और OTC in any medical shop. But before that, excuse me, our director is calling. Just a moment, let me. सर मैं वो आपकी लेक्चर दे रहा था, काफी मैं दे रहा हूँ सर, लोग 5 मिनट बस. Sorry. So in fact I tell you there is a word reverse pharmacology. Reverse pharmacology is best suited for any plant-based product development because we have traditional knowledge available with us. In that traditional knowledge or traditional text, it is well known for a particular disease or disorder. So what is required? So the requirement is very clear that you need to validate those traditional claims. So during the validation or during that research activities, sometimes we get new leads or novel leads by which we develop some different products. But in reverse pharmacology, it is very simple way to validate a product or to have a kind of data by which you can go on global competency. So what we do generally, in general, we follow the traditional approach. We consult all the traditional texts. Then finally we select the potential species. And since we are, as I already briefed you about the sources or location specific sources, so we go for the best sources, we collect the best or potential sources, and then as per guidelines, as per SOPs, we go for its extraction, fractionation, or whatever is required. In general, just I wanted to share one of my own experience that in if you are going for developing a kind of any product, whether it is for any wet product or even termites, even we have also a few products in that area, and the most important issue is which extract you are trying to validate. So here my experience is and my submission to all that you first try to evaluate or validate water extract and then hydroalkali 50-50, and then thereafter you can go for any extraction protocol or even fractions or subfraction. But ultimately what will happen if you go for the fractionation, if you go for the enzyme extract or any subfraction or even isolating the pure molecule, you will end up with zero, because sometimes pure molecule is having good efficacy, but in most of the time, in case of medicinal plants, it works in synergy. So if you are having a kind of combination of different species or combination of an extract, it would be better for developing good quality product. So extraction, fractionation, then activity guided isolation of active compound, then validation, which is pharmacological studies. It's a pre-formulation studies. Then you have to develop a formula, its dosage, then post-formulation studies for standardization of the compound formulation which you have developed. Then it's safety and toxicity issues as per OECD guideline, and then clinical studies as per DCGI norms. Now in DCGI also we are having Ayush cell for our Ayurveda based products. Earlier it was not there, but now we have a Ayush cell in DCGI, Directorate General of India Office, FDA 1, Delhi, where you can get approval of clinical validation for any of your product. And although as per regulatory guideline for any Ayurveda product, clinical validation earlier as per Drug and Cosmetic Act was not essential. But for global competency, you have to have all sort of data. Without that data, you will not get opportunity for global positioning of any of your product. So that is why the government has established one Ayush cell in DCGI, because as per guideline, which we sometimes the guideline itself is misleading, that not required, clinical trial not required. However, it is very clearly written that clinical trial is not required for those formulations which is already established and documented in any of our traditional text or AFI, Ayurvedic Formulary of India. तो कभी कभी हम इसके आड़ लेकर के हमारी इंडस्ट्रीज जो इसकी आड़ लेकर के वो क्लिनिकल ट्रायल नहीं करते हैं, छोटा सा अपने प्रोडक्ट लिख देते हैं, नो ह्यूमन स्टडीज, नो एनिमल स्टडीज. But now, if you are going for global competency, you have to have all sort of data. And of course, when you do all these things, you can protect your IP, and your final product is ready for global positioning. So these are few of the examples. Dr. Tarun has already given an elaborated list and of product as well as my candidature. So these are few of the examples which we have successfully commercialized and earning a lot of money for our institute. This is another product which we have recently launched, Euro 5, which was initially claimed for kidney stone disorders, and now getting a lot of attention along with its urolithiasis other targets also, because we are getting feedback from urologists all across the country that this is really a good product for some other targets. So we are also trying to improve and modify it and going for another patent for out of it. Dr. Tarun was mentioning about that the herbal immune booster Josh. These two products are need-based intervention. So our scholars, those who are listening here, even our scientists or young researchers, those who are here, I suggest need-based intervention. This word particularly was effectively implemented during COVID और फर्स्ट वेव ऑफ कोविड. हमें कोविड से उस समय इतनी दहशत थी कि हमें कुछ पता ही नहीं चल रहा था हमारे साथ होगा क्या. जो भी जो बोलता था, किसी ने बोल दिया काढ़ा पी लो, किसी ने बोल दिया कि लौंग खा लो, कोई बोलता था कपूर सूंघ लो, हम सब करते थे. कभी ये नहीं सोचा कि इसका एक्सेसिव यूज हमारे लिए कितना नुकसानदायक होगा. जितना पोस्ट कोविड इम्पैक्ट हमारे कोविड के इन्फेक्शन का है, उससे कहीं ज्यादा हमारा अपना खुद का लिया हुआ सप्लीमेंट्स है. लाइक जस्ट आई एम शेयरिंग वन एग्जांपल दैट हम लोग खूब काढ़ा यूज किए. वी हैव यूज्ड अ लॉट ऑफ काढ़ास. घर में इमोशनल होते थे. माँ बोले थोड़ा सा अदरक बढ़ा दो, पत्नी है किसी की बोले नहीं नहीं काली मिर्च बढ़ा दो, हमारे पति की गले में खराश ज्यादा है. किसी की बहन बोले भैया को सर दर्द हो रहा है तो उसमें मुलेठी बढ़ा दो. So we were not having a proper recipe for preparing such kind of products. So in terms, what was happening is, or not only dosage form. So what was happening? किसी को घर में बहुत ज्यादा ओवर कंसर्न है, 2 बार काढ़ा पिला दो, 3 बार पिला दो, कोई डर के मारे 5 बार पी रहा है. तो उससे होना क्या शुरू हुआ? Lot of issues happening for acidity, ulceritis, pancreatitis, and many such GIT related disorders or disease. So at that time we were already in touch with many agencies, even though lockdown was there. So we thought to make a optimal dosage and a recipe or a product which will be uniform and without any harmful effect. So we started working on this kind of kadha. So what we did, it's a recipe of few medicinal species, and then what we did, we have changed its dosage form. It was simply need based intervention. कोई हमने कोई डिस्कवरी नहीं की, कोई ऐसा हाई एंड रिसर्च नहीं किया इसमें, और नीड बेस्ड इंटरवेंशन था. हमने इसके डोसेज फॉर्म को ऑप्टिमाइज करके इसको 2 ड्रॉप्स पिलाया. 2 ड्रॉप्स 40 mg ड्रॉप्स, जो हम लोगों ने लैब में अपने वैलिडेट किया. वी हैव एनालाइज्ड प्री एंड पोस्ट फॉर्मुलेशन स्टडी, एंड देन with approval and getting CTRI clearance, we proceeded for the clinical validation through one CRO. Surprisingly, this product is highly effective in cough, cold, and flu-like symptoms, as well as in mild COVID cases. We have already approached Ministry of Ayush for its endorsement. It's in process. But this product got attention not only of our frontline warriors, but as well as to our government officials and PMO. So that is why we transferred. We were in pressure that for its supply. So we were initially serving to our local frontline warriors, but then when it was in demand, so one of the company, this Mark Laboratories Private Limited, we have transferred this technology to them, and now it is from this season it is available to common public in our country. Another one is this mask herbal decongestant spray, which Dr. Tarun was mentioning about. When you Google it, herbal decongestant spray, you'll get all the details and about the product as well as our claims. What we did, nothing new we have done. It's only an out-of-box thinking. It's a combination of 3 oil in a fixed ratio and single shot spray on your mask, in your room, on your pillow, will serve the purpose of choking, suffocation, and even have this ease out through these 2 choking and suffocation issues because of usage of mask. सो हम लोग मास्क लगा के बहुत घूमते थे और उससे बहुत प्रॉब्लम होती थी। सो आपको इसको यूज करने से बहुत इफेक्टिव हुआ है, और हमारा सबसे बड़ा इसका जो प्राउड फीलिंग है कि हमारे जो मेडिकल स्टाफ था उस वक्त, whether it is from our King George Medical University or even in other hospitals, we provided them, and they were using a lot of masks that time, and we have feedback from and proof of use from them, where we have appreciation for this kind of product. Even our officials of state of Uttar Pradesh, they have also given testimony of it. And then finally, we proceeded for evaluation of nasal edema, which is happening because of congestion and choking. So in that case, even it is really very effective. So what I mean to say that strategic product development and then need-based intervention, these two parallelly you can think of. And it doesn't matter whether you are from forestry background or even from botany background, even from chemistry background, even not from pharmacology background, even though you can work on it. You have to make a team, you can complement, or you can get supplement from other experts of your area, even from after you itself you'll get a lot of backup. Dr. Tarun is with us in several committees, and he is really a gem with you, and he's having a lot of experience in this area. At least with us in team, we have already seen in his input in different committees. So need-based intervention also, wherein you can focus solution. So this is that product. I was talking about the Ageratum conigeatis that time, and this is the product which is now available. And this is a product not only at global level, first of its own kind, we also got appreciated and recognized from ICR, and ICR has given a kind of appreciation to us that this is their this is the best product, or ICR this is the best product. It's a patented product. Jointly we have developed with IVRI. Product formulation was our job. Validation was for IVRI. So this is an example of consortium mode product development, wherein we jointly worked and we have delivered. These are few other examples of our inventory. Now coming to the last slide, what not we have. I told in the beginning, yes, we have rich bioresource, we have rich ancient knowledge, we have modern science with us. All these three can transform, and you may have herbal medicine. Nowadays, phytopharmaceutical is emerging, nutraceutical, of course, and then cosmeceutical products. And all these products leads to health security, economy, and employment opportunities in our country. So it's a kind of chain reaction, start from field, from forestry background to chemistry background or even management background. We all have opportunities for global positioning of our own traditional heritage, and for our own proud feeling that yes, we are working for our country. That's all with this. Thank you so much. With this line from Swami Vivekanand, that arise, awake and stop till not the goal is reached. उठो जागो और तब तक नहीं रुकना जब तक कि लक्ष्य हासिल ना हो. Thank you so much. Thank you all for giving me this opportunity.
busy schedule. And so we have with us Dr. Sharad Srivastav.